Welcome to the Catalysts podcast. I'm your host, Patrick Morley. What if the key to improving cancer treatment lies in the trillions of microbes that live in your gut? Today's guest, Dr. Saman Maleki from the Schulich School of Medicine and Dentistry, is leading innovative research that uses the microbiome and even fecal transplants, or FMTs, to supercharge cancer immunotherapy. His work is a story of how persistence can turn skepticism into progress that is changing the way we think about modern cancer treatment. It's a pleasure having you here today, Saman.
It's a pleasure to be here, Patrick.
Yeah. Our gut and its complex world is clearly having a moment in 2025 with all of the amazing scientific discoveries. But you've been interested in this field for quite a while now. And for those of us who aren't scientists, can you paint a picture of what is going on in our guts and what that could mean for cancer patients?
Yeah, sounds good. So, I've been studying the microbiome for a while now. But even as a grad student here at Schulich, when I did my PhD, I was always fascinated with how the immune system in our body interacts with all these organisms that exist in our gut. In fact, the biggest immune organ we have is the gut, because you have all these trillions of bacteria in the gut, and they're constantly in crosstalk and conversation with the immune system. And you need a very well-balanced homeostasis. That means everything's going very well in this conversation. Nobody's getting really angry, all the immune cells, all the police in our body, to keep the health and bring down the inflammation that can arise from an angry immune system. And then with some conditions, some chronic conditions, some diseases like cancer, for example, the balance in the healthy microbiome, those good bacteria that exist in our gut, that they're functioning day in, day out, producing all the metabolites, molecules that the rest of the body needs, can go off a little bit. And what that do is that it can make the immune system angry. You can experience inflammation. It can actually cause the outgrowth of some bugs that they will help the cancer in many ways by helping suppressing the immune system. And that's why gut as an organ is the one that in the era of immunotherapy that we can potentially change and manipulate to improve the immune response that we are so desperately trying to leverage with these new drugs that we have in the field called immunotherapy.
So the gut is an organ.
Yeah.
In the body, right? And a lot of people don't really understand that.
It is, I guess, an ignored organ. It is an organ for itself. And by organ, I mean the gut microbiome, the bugs, those trillions of the bugs. We have to treat them as an organ, really, because they live with us throughout our life. Obviously, they're not directly part of the host, but they are there and they contribute quite a bit to our health. The microbiome, I like to think of it as this big pharmacy. And it's producing a lot of different metabolites, a lot of different molecules that are helping different organs function properly, even in your brain. And now there is this gut-brain axis, and there's like molecules that are produced in the gut, but they make their way into the brain and help with the function of healthy brain. And now imagine if you have, because of aging, because of inflammation, because of chronic conditions, because of things like type 2 diabetes or other conditions or cancer, if you shift the balance, if you shift those healthy microbes in the gut, you're going to effectively contribute to the downfall of a lot of important functions in the body. And one that's really, really important for the fight against cancer is the immune system. And we know the gut is regulating, talking to the immune system constantly. And now if you don't have a healthy microbiome, there is this real possibility that the immune system is not doing what it's supposed to do because it's getting all these wrong signals from the gut. And we know there are studies that show cancer is editing the microbiome, is shaping the microbiome the way it's wanted to go. And the microbiome becomes a supportive organ for the tumor to grow by suppressing the immune system. And that's why it's so important to have a healthy one.
So in this pharmacy of the gut, it sounds like there's a little bit of a power struggle happening when cancer enters the system. Mm-hmm. Is that a fair assessment?
I would say so. Cancer is a really interesting problem because as it starts to grow, it's actually growing against very difficult odds, because in our body, we have a lot of systems in place built into the cells and externally with the immune system and everything that's trying to find these transform cancer cells and destroy them. And those mechanisms are very successful overall. That's why not all of us are dying from cancer all the time.
Right.
And but despite the fact that cells transform in our body, become cancerous, but they actually been picked up or they die of the mechanisms that involve them. Now, when the tumor gets a foothold and becomes an established tumor, it's now starting to invade the surrounding tissue. It's starting to create factors that is creating all this mess in the body. And one of the organs that's been affected is the gut, gut microbiome, because it's getting all these signals from the tumor. And that power struggle is happening with the tumor trying to be the dominant factor here and trying to effectively not only affect the microbiome, but also the immune system. We know parts of the immune system become tumor supportive,
Right.
and they actually home to the tumor and support the tumor against the rest of the immune system. There is this really fascinating battle going on inside the tumor, but it's a part of a bigger war that cancer is waging in our body against our organs, which is, evolutionary speaking, cancer is not very smart because it ultimately kills the host. And by killing the host, it dies itself.
Right.
It's like a parasite that's growing. It's not a successful parasite that keeps the host alive because it just wants to become dominant and dominant and dominant.
Right.
And at some point, the host will fail, the organs will fail, and the cancer dies with it.
Right.
So that's the problem.
What does it mean to have a healthy gut?
Mm-hmm.
I think everybody's gut is different. I mean, there's trillions of microbes in there. Is there a benchmark as a researcher that shows, okay, this is a healthy gut, this person maybe doesn't have a healthy gut, or this cancer patient's gut is below?
Mm-hmm.
Is it a matter of the amount of microbes? Is it how they interact with each other? Is there a benchmark to know that?
That's actually a really important question. I think one that a lot of different scientific groups are trying to address. And I got to say, with all the progress we've made so far, I don't think we have a unifying definition of what's a healthy microbiome. We have some rough measures. For example, excuse me, like diversity is an important factor. So you're having different kinds of bacteria in the gut. So, and you don't have one bacteria or two bacteria becoming dominant. It's like, think of it as a successful functioning society.
Right.
In a society, you need teachers, you need police officers, you need doctors, you need nurses, you need judges, you need everybody.
Yes.
So now if you take a few of these groups out and replace them, let's say, replace everybody by doctors, do you think that would be a functioning society?
Right.
No. Who's going to come in and fix your pipes if your sink backs up?
Right.
You know what I mean? You need a plumber.
It's an argument for diversity, right?
Exactly.
Yes.
So, and we see in nature, diversity is actually a really important strengthening factor. And one of the things that we lose naturally, actually, with the process of aging that has been shown in the field of microbiome is the diversity in the microbiome. And some centurions and people who actually age very well, when they sample their microbiome, it looks like they have a very diverse, younger looking microbiome compared to their other people who are in the same age category, but they're not doing that well. So, and I guess that is one of the factors that we look at. So it's not true for all cancers. There are some cancers that the patients, when you're sampling the microbiome, you still see there is a diverse composition, meaning there are different bugs that you can identify.
Right.
Now, when it comes to the function of those bugs, that might be a different story. And that's where the field is going. And that's one of the areas that my group is pushing forward, is that not just looking at the composition and the makeup of the bug, but their function. You do have the plumbers, but when you call that plumber, is the plumber able to come in and fix the pipe, or it says that, sorry, I'm just not available?
Right,
so
right.
That is what I think we're learning that might be missing in many cancers.
Right. Can you take me to the origins of your fascination with this, and sort of what inspired you to think of this connection? Can you take me to that moment and then the journey that you've had to take, which I know has been long and it's been arduous, but your persistence has really paid off. Can you take me there?
Sure. So I actually got my PhD here at Schulich in 2014 in the Department of Microbiology and Immunology. And I was going to do my postdoc somewhere else. But at the same time, I was looking and getting very interested in what was an up-and-coming area in the field of immunotherapy, and that was a potential role for microorganisms. That when I was looking at the literature, I was seeing that there is a possibility that the microbes are doing something in our organ. These are early days for immunotherapy with checkpoint inhibitors, these antibodies that they have been used in the clinic extensively now to treat the patients. But there were some preclinical studies, meaning mouse studies, that was showing that the animals that are responding well to the mouse version of these drugs have a particular version of microbiome, or some early observational studies, meaning patients were getting their treatment, but they're getting sampled, and people looking at the microbiome and saying that those who have a more diverse, healthier looking microbiome are doing better. So it was giving me this signal that there might be something there, but it was very, very early. And given the fact that we have different groups here in London and part of Schulich, that they do microbiome research, and they do actually microbiome research in humans. We have a strong oncology group. I actually decided to stay around and poke at this a little more. And by looking at it more and more, I was convinced that there is a role for the microbiome. But also, I think an opportunity that I saw with the microbiome at the time was very difficult for most people to see, was that it's a malleable organ. You can shift it. You can change it. It's not like necessarily like your bones. Even your bones can change. You know what I mean? But it's much harder, because we know that we can shift and change the microbiome with diet, with exercise, or with antibiotics. And also with this process called fecal microbiota transplantation, or FMT, which I knew was going on here in London, not for cancer patients, but for other groups of patients who have C. difficile infection. It's a common infection.
Which we'll talk about it a little bit later,
Yeah, so, and that got me interested in a possibility that we might be able to prospectively change this in patients. But this is like around 2015.
Okay. So about 10 years ago.
Yeah, it's about 10 years ago now. And this was a very different arena. People would not be open and receptive to this idea, especially in oncology, where you're getting oncologists starting to understand and treat their patients with these new drugs, immunotherapy. At the beginning of this time, even most oncologists would call these drugs just chemo, because they thought it's just similar to chemo. And it took some time for even them understanding these are not chemotherapy. These are actually immunotherapy drugs that are not
living cells, right?
So living cells are the CAR Ts and cell therapy, and those are true as well. And microbiome plays a role there as well. But for even these antibodies, they're not directly targeting the tumor, but they're actually activating the immune system.
Right.
And what I was going after was that missing factor one level below this, where you're thinking about the conversation between the immune system and the microbiome. And I was thinking that a healthy microbiome can help effectively get these T cells in a better place when they see the drug. Now they can go after the tumor. And that was not very popular at the time, I would say. One, because the evidence in the literature was sparse. And the other thing was that to just try something like this in cancer patients, where oncologists are warming up to immunotherapy, and now you want to add another treatment that might actually now activate the immune system even way more, was very, very difficult. So it took me some time and some convincing of colleagues and different meetings and conversations with different groups. And the other part of this is that this would have required a very multidisciplinary approach. Today here at Schulich, we are probably one of the best in the world. But back then,
at collaborating,
for this particular area, I can say we were not. Nobody was. So, and that was one of the reasons nobody was really interested in this, because you had to go bring infectious disease doctors to the oncology clinic to effectively work with them, with the oncologists, with the immunologists, with bacteriologists. You need a truly multidisciplinary approach to be able to deliver a treatment like this. As simple people see it today, it wasn't like this. So, and that was also a big, I would say, barrier. So that I actually took it upon myself to try to solve it by initially meeting separately with the oncologist, trying to explain the rationale for this meeting with the infectious disease teams, with the bacteriologists.
Right. Take me to to those conversations. How, like, you would explain this idea, and would they kind of start looking off in the distance?
Yeah, so I gotta say, so it was different groups had different reactions to it. When I went to see Mike Silverman, who's the chair chief of IED, who's a very good partner of mine today, and we do all the FMTs from his lab that they manufacture the capsules and work together very closely today. When I talked to him about a possibility of doing FMT in cancer patients, and back then they were not even doing it in using capsules, they were just doing it using colonoscopy and enema.
Right. And much more invasive and expensive.
So, and he was like, you know what, I don't remember any time like somebody from oncology would knock on my door and say, we want to use some of your tools for treating our patients. This is really, really odd, but sounds interesting.
Right.
So, but I'm not the person you really need to convince. It's the oncologist that you need to talk to, because those are not my patients. So I took it that he's interested in this. I did tell him at the time that it might be difficult to do this with colonoscopy and enema. Nevertheless, I had to persist to see what we can do. And then went to the oncology team. The conversations there was quite different, actually. So I some of my very good friends and colleagues today, when I would pitch them the idea for the first time, they would literally just walk away mid-conversation.
Mid-convo,
yeah, just walk away. They would think that this guy is completely out of his mind.
Right.
So, and it took quite a bit of time, quite a bit of efforts. You were
leaving papers behind,
so
like in your wake.
That we had a melanoma disease site meeting that I started to go to these disease site meetings.
Yeah.
And one of the reasons for it was that I realized that despite the fact that I'm in the cancer center, our lab is there, not all of these people actually know me. So, and they may feel that I'm an outsider, and I'm trying to invade their space. So I decided to actually attend their disease site meetings, try to learn and understand their problems, their concerns. So, because a lot of times it's also very important for you to understand what the other person is thinking about this, what are the barriers. So, and I remember when in one of these meetings, so I wouldn't really given an opportunity to speak, but what I did was I printed a paper I knew, because we were getting email lists, how many people are at that meeting.
Right.
And I printed those that was about these advances about microbiome, understanding about the microbiome in particular in melanoma. And I left a copy for everybody at the table before they actually show up. I went there early, and some of them were surprised to see this thing. And I think it got through maybe one or two of them that they actually read it and actually made the conversations a little easier.
Yeah.
So, and it was also from the aspect of you can't do a trial if you don't have funding. So I started to look into where I can actually submit an application potentially to get a grant for this. And I can tell you, nobody would give me funding for such a thing to do.
Yeah.
Then one of my colleagues told me that one of the foundations out west might have a call for alternative medicine for oncology at the moment. And I really appreciate these guys, actually, is the Hecht Foundation.
The Hecht Foundation, okay, yeah.
Yeah, so I called them up, and I said, I have an idea that may be alternative enough to fit this category. And they said, the grant officer asked what was the idea, and I said, I want to treat cancer patients with poop. Right. So do you think that's alternative? And they said, yeah, that sounds pretty alternative to me.
Definitely. Yeah, you fit the bill perfectly.
Yeah, so they told me I can write a grant. Yeah. I ended up writing a grant. I had nobody from the oncology team to actually even read the grant at the time.
Right.
I wrote the grant. I sent it to them. They liked the grant. They got back to me and they said, we like the idea and we think this is quite remarkable if you can do this, but it's very clear that you have no oncology support. You want to do this in patients, but if you have to have at least one oncologist.
Yeah, just one, please. Like, one would be nice.
So, and then what happened was I went to John Lennon, one of my very good friend, who is one of our oncologists here at Schulich, who treats melanoma. I've been constantly talking about this, and I guess he was maybe crazy enough to agree to be part of this idea. And he read the grant and he made a couple of corrections, and I sent it back to the foundation, and they funded it. So they funded it, and that actually really now jump-started this this field for us in Canada, not just for us here in Schulich.
Right.
So because what happened was now we had funding, and I had to now write a proper, protocol for the trial, go through ethics, all that stuff. So and started to do all of this. And I was trying to learn. Remember, I'm a trained PhD scientist. I'm not a clinician.
Right.
Today, I know a lot about clinical research because that is the majority of things that my lab does.
Yeah.
But at the time, I was just trying to read. I was like trying to go to clinicaltrials.gov and see similar clinical trials that was happening in melanoma patients, not necessarily with microbiome, but like what they are getting, what's their exclusion criteria, and then bugging John with it and say, do you think this would fit or this doesn't fit, whatever. So trying to put this together.
Right.
And then ultimately, we had a protocol, and it took us some time to actually open the trial from the original idea and getting the funding. I think we opened the trial in July 2000, I wouldn't say 19.
Okay.
So
roughly 5-6 years ago.
Yeah. So, and to get the first patient, and we also were, I was able to work with Mike and his team, Dr. Seema Parvati, who's a great collaborator and partner in this, to actually not do this through enema and colonoscopy, which would have been very difficult. So, and they actually switched to capsules, and we also helped them to develop the capsules. So, and that actually also helped advance the FMT field on its own. So what happened was we ultimately got our first patient on the trial, treated her with the FMT capsules, and then she got immunotherapy. I remember the day that happened. I have a picture of me holding those blood samples from the patient, bringing it back to the lab. But I went to John's office and I said, either we opened an avenue in oncology today, or we're just going to kill a field if this goes really bad for the patient. So obviously we had to do a safety trial.
Yeah.
So we did treat the first patient. She actually did well in terms of safety-wise, and she had also reasonably good response to her cancer treatment. We treated the second patient. It was just a gentleman who had quite a bit of melanoma metastasis in his lungs, and he had a complete response. That means all the tumors melted away. We treated the third patient, same thing. All the tumors gone away. The fourth patient did really well. So at that time, I put an abstract together and submitted to SITC, Society for Immunotherapy Cancer, a late-breaking abstract submission, and got accepted as a late-breaking poster. And I went to SITC and presented that. And that's when actually my phone started to ring, because people said that, okay, you did this really out-of-the-box idea, and it looks like the first 4 patients that you treated, they all did really well, and nobody had any major toxicity.
Right. So in such a short time, I mean, it might have felt like a long time to you, but in such a short time, it seems like you went from colleagues, respected colleagues, friends, you could call them, walking away from you, or, you'd be banging on their door and they're not answering, to actually having clinical trials and successes. Like, how does that make you feel? I mean, it's really a story of persistence.
Yeah, I would say it took from the ideation 2015 to the first patient trial 2019 some time. But even after that, we did face quite a bit of challenges because COVID hit obviously in 2020, and that was the time that we opened our second trial perform in kidney cancer patients, and still couldn't get a lot of funding. Actually, the London Health Science Foundation and donors supported that trial quite a bit, which I'm forever grateful. But we treated the first patient, and then we had to halt the trials because of COVID. And then it turned out that the virus was found in stool. And now Health Canada put us on notice, saying that you can't do this until you can actually figure out how you can screen the donors for this. And we were actually on hold for about 6 months. I had to work with Mike and Seema to try to develop screening methods for donors with FMT. I started to look around and see which lab in the world actually does COVID screening in stool, and it was no lab. This was a new pandemic. So, and then we worked with a lab in Georgia, in Atlanta, that they started to do, is a CLIA certified lab that they started to do in stool testing and send samples then, but they had a lot of false positives, and then we had to switch to a lab in Toronto later on. But after 6 months, we were able to publish a paper showing that we were actually able to screen the donors and the stool, and which, by the way, we are still doing it today. That hasn't changed despite the fact that COVID has become more of an endemic problem. And then to convince Health Canada this was safe, and actually helped everybody else in the field. So I got to tell you, I think the effort I put into the process of this, which helped the patients, also change people's ideas, because also they are seeing it in their clinic when the patients are helped. Now, most of the trials we did early on were safety, non-randomized one-arm phase 1 trials or phase 1/phase 2 trials. These are smaller trials where you don't have another arm of control, where you're comparing your treatment to the standard of care to prove definitively that this is the FMT that's doing it.
Right.
But that's something we are doing now.
Okay.
So we're doing now in large randomized trials of melanoma and lung cancer to prove that this is the treatment that's doing this. And if those trials are successful, then we go for phase 3 trials that they are international trials, and they will help us hopefully to change the standard of care for those cancers. But I guess what we also tried to do, and I was being a scientist and being a trained immunologist, but it was also important for me, which effectively you need in a scientific community, is that to do good science. So we did treat the patients. Obviously, you're getting the clinical signals. But what we also did was we analyzed the microbiome. We actually showed that every patient in that first trial who did really well in response to FMT and their immunotherapy treatment were the ones that they had a successful engraftment of the donor microbiome. So the donor microbiome actually went into their gut and stayed there for at least 3 months, as opposed to the patients who actually lost the donor microbiome, went back to their own original microbiome, and they did not respond to treatment, to their cancer treatment.
Right.
So we created scientific evidence that also supported this notion that the microbiome is really important. And then we showed also the effect on the immune system, the changes that was happening on the immune system.
Right.
So
you're piling up the evidence at this point,
yes,
right?
So you need not only the clinical evidence, but also the scientific translational evidence around this to convince the scientific community that this is working through the means that you're saying it's working.
Yeah.
And that allowed more people to be interested in the field and more people trying to do this. And I can tell you that after we published that first trial in Nature Medicine in 2023, I was told by a whole bunch of leaders of the field that actually rejuvenated the field of microbiome oncology. Now, there's a lot of trials that are launched around the world that people are trying to follow our protocol and try to do this thing. And because they saw it was good science and was good clinical data. And I think that's really important. You need, in the world of science, you need many, many groups to reproduce your findings,
right,
to show that this is actually true.
The hallmark of the scientific method is reproducing results, right?
Not just one lab.
Right.
Yeah.
This mini-series is in support of the J. Allyn Taylor International Prize in Medicine.
I'm really curious if you could take me through a fictional case of, a patient has seen their family doctor, they're not feeling well, or it's a routine blood work shows that there's cancer. Obviously, pretty shocking news. How does a patient go from there to ending up on your radar in a clinical situation? Can you walk me through what that looks like for patients?
Sure. So we have done most of these trials so far on patients who have advanced and metastatic cancer. These are patients whose tumors are too large or too big to be cut by a surgeon. They call them locally advanced, or patients who have a number of metastases. The tumors have spread throughout their body. So these are advanced cancers.
Just like stage 4?
Stage 4 patients, mostly stage 4, but some stage 3s as well.
Okay.
So these patients come to the clinic to see their oncologist. And what we have been able to do with our group has been to treat these patients in first-line setting, meaning before they actually start their immunotherapy treatment. And that was a very critical point for me when I designed these trials initially. And a lot of times when you're going with a new drug treatment, because you have to establish really safety, and it's like some of these chemical drugs and stuff, you're only allowed to treat the patients after they've gone so far down the line of treatments. But that, I thought it was a detrimental factor to a microbiome therapy, where if we were able to get the patients first line, treat them
earlier
before they go and burn their immune system through different treatments, we might have had a better response. There are other groups that they have treated patients who we call refractory, that they have treated, they received immunotherapy, they didn't respond to the treatment, the tumor didn't respond, and now they're giving them, let's say FMT, and then treat them again with immunotherapy. And some of those patients actually show response, which is great, showing that microbiome actually works even in a later setting, but the results is not as strong in terms of clinical response as what you would see potentially in the first-line setting. So we were able to get Health Canada or convince Health Canada to allow us to do this first-line treatment because FMT generally is safe and it's actually standard of care for C. diff infection in patients in Canada. Yeah. So not cancer, but it's not an unsafe treatment overall.
Right.
So long story short is that the patient gets referred to their oncologist. And what we have is that we have different cancers that we treat in different clinical trial settings. We are not allowed to treat patients outside the clinical trial setting, although I get quite a bit of requests, believe it or not, from not patients in Canada, but internationally from everywhere who ask for this treatment. And then they get screened by our staff in the clinical research unit at the London Health Sciences Centre, at the Verspiden and Family Cancer Centre. If the trial is open only in London, if trials open in different centres or multiple centres, they also get screened at those centres. And if they actually qualify, that means they meet the eligibility criteria, they can be treated with this treatment. And then the CRU member or coordinator goes and talks to the patient, and the clinician in the room talks to the patient, and then the coordinator goes talks to the patient about this new treatment. And if the patient is agreeable to it, they give us consent. Then effectively they sign in to be part of a clinical trial or on this ongoing clinical trial. And then what happens is that now they are treated as part of a clinical trial. They have to effectively follow the clinical trial protocol. That means they may have to come in to give us more samples, more clinical samples like blood, stool at different time points. For FMT, what we do is that we also notify the team at St. Joe's where the capsules are coming from. And then what happens is that for some of the patients, we actually send them to St. Joe's, or for some of them, we actually treat them at the Verspiden Family Cancer Center, depending on the cancer.
Okay.
And then, so the patient actually undergoes a bowel prep, standard bowel prep, the night before FMTs. This is similar to what people do before colonoscopy.
So cleans out the system.
Cleanse the system, bring the load down and the microbiome load down. And the rationale behind that is that you want to make room for the new microbiome to come in and be able to stick.
Right.
If you have a lot of your own bacteria, what they do is that they outcompete, because they're established already, they outcompete the new microbiome. So you can't really give somebody a successful FMT if you do not get rid of as much as you can of their native microbiome. Right. So they do undergo the bowel prep, and we ask them to effectively make sure that they have a clear stool before they come to the clinic. If the stool is not still clear, that's we actually don't do the treatment.
Okay. Is it fair? Is it accurate to say you're literally cleaning out the pharmacy?
Exactly.
You're cleaning it out so you can restock it.
You're sending the old staff away.
Yeah.
Putting them on holiday.
Yeah.
And then what happens is that now you're effectively bringing new staff, new pharmacists to go in that pharmacy. So they come to the clinic, they either as Verspiden Family Cancer Center or St. Joe's, depending on the trial. And then we give them about 35 to 40 capsules. These are 3-layered encapsulated capsules. They're
quite big.
Yeah. So, and they have about half an hour to take all of that. And that gives them about 100 grams of stool that we actually get initially from a donor and process. That would be a big enough dose of FMT that would effectively almost guarantee replacing the biome with a new biome. We actually give patients a bit higher dose than most other FMT studies.
Okay.
So, and I think that has been an important successful factor for us, and I think other centers now trying to do that as well. So, and 40 capsules sounds quite a bit. That's true. Yeah, but I got to say, as humans, I think we all get really good at taking capsules as we age, and most cancer patients are older individuals, and we haven't had anybody who actually has had problem taking the capsules. So they're just sitting, they get a sip of water, they have a few capsules, and So, and then you have to take the entire capsule. So because that's the full treatment.
Yeah.
Most of the trials, that's the only treatment we give them. In one of the trials that we have now under review in the journal, we actually gave them 2 additional doses of FMT as well.
Okay.
So, and then what we do is that we tell the patient that go home, you have to wait at least about a week So, and that week time is the time that we allow that new microbiome or those new pharmacies to actually take a foothold in the pharmacy and start producing.
Yeah, they need to know the protocols, get trained.
So we don't want to start the cancer treatment right away, immediately, because we want the pharmacy to start to work to the microbiome to start producing everything that helps the immune system, and now go treat the patient with immunotherapy drugs that are going to boost.
It's like a soft launch for a restaurant, right? You get it in there, they get trained, they get comfortable.
Yeah.
Then you open up the doors for the immunotherapy.
Exactly. Right. That's what we do. And that has been a sweet spot, I would say, that we had to find for this, because in oncology, you cannot really delay cancer treatment as much because it's a life-saving treatment.
Time is of the essence.
But also we knew from all the studies that were done, especially preclinical studies in animal models, that we do need some lead time to let the microbiome take foothold if we want to have the best outcome. And the way we arrived at that was also interesting historically, and this is pretty much now standard for most FMT trials. People use 7 days, but the way we arrived at that, which is now adopted by most other trials in the world, is that we actually set up a meeting for this particular topic at St. Joe's, and I went there with John Lenahan and another person from the oncology team, and we meet with the FMT team there with Mike Silverman and Seema, and we're sitting at this table, and this conversation about Mike says we need time, maybe 2 weeks. And John says, no, we can't wait. We have to start treatment. And they're looking at me and I say, 1 week, guys, 1 week. It's like, it should be enough time to get the patient prepared. They're going through the process at 1 week, and then that should be enough time for the microbiome to take. Yeah. So it wasn't exactly science, but it was more being pragmatic and finding a time that works for both teams, because we're doing a multidisciplinary trial. Yeah. So the infectious disease team is thinking about what's best for FMT and that new microbiome to take. But the oncology team is thinking about what's best for the cancer patient and the treatment with immunotherapy.
So you had to meet somewhere,
and that's where this is an example of one of those challenges where you're working a multidisciplinary trial and approach in science. You have to understand where everybody's coming from and try to address. You can't just say, oh, I will not hear you, and just say we have to do it this way. No, you have to try to address that to the best that you can. And in fact, our studies in that trial and our trials showed that 1 week time was enough for the microbiome to take, and that has become pretty much standard. So I was asked in a conference that, why didn't you wait 2 weeks? The mouse data is showing 2 weeks is better. Yeah. And I was like, I'm not treating mice. So you go tell that to the oncologist. It's not as easy to tell the patient, I'm going to delay your cancer treatment for 2 weeks. They are very anxious to get their cancer treatment started. Yeah. And we had patients, maybe a couple, who declined to go on the trial because they didn't even want to wait 1 week. Yeah. And that's understandable. So although we think that if we give you FMT, you overall you'll do better.
Right.
So, but that's what the patients will experience. And then they start their immunotherapy, and along the way we will collect samples from them. And one of the things that we did was we built this new treatment on the back of standard of care, meaning we didn't want to change their cancer treatment, their standard cancer treatment as much, because that would have made it very difficult for the patients, and then it would have required much more funding from us, and convincing drug companies, which is very difficult to do. Right. So that's why we tried to just get the treatment initially and then let the patients continue on their standard of care journey, and we're just collecting samples from them and looking what's the change that's happening to their microbiome post-treatment, what's the change that's happening to their immune system, what's the change that's happening to their metabolome. Because the analogy of the pharmacy is that these metabolites are produced by the bacteria, and you can actually detect those in the blood, in the stool, and sometimes even in the urine. And we are actually testing for those and seeing that, okay, now we did change the microbiome, and we are seeing downstream of the microbiome, the products that they're producing, we can detect them in the blood of the patient who's responding really well. That's what we expect to see. So we did continue that, and we continue collecting samples for different time points because we want to see longitudinally how long this new microbiome will last in these patients. And does it correlate? Does it actually show that if they hold that microbiome successfully, they are responding to the cancer treatment or not? So this is what the patient could expect to go on an FMT trial.
Right. Thank you for that explanation. It makes a lot of sense to me. this is just curiosity that popped into my head. During that week where they've taken the 30 or to 40 pills, does that patient feel different?
Oh, that's a wonderful question.
I mean, you're literally, like you said, you've booted out the old staff. You've got new staff in the gut, which is responsible for, we've discussed many things. Are there reports of people feeling different or having
I got to make
mood
sure
changes?
That you're not working for Health Canada, are you?
So I am not.
That's a wonderful question, actually. That's a very important question clinically, also a very important question scientifically, one that we have to address. So because we have that 1 week window, you're absolutely right. The patients do experience something. So, and we actually track it. So we actually want to see if they're experiencing any side effects of the treatment. And because that's a window that they haven't got immunotherapy, we can directly attribute that to the FMT. And as a matter of fact, most patients are experiencing what we call grade 1 or 2 self-limiting toxicities, like mostly GI-related toxicity, something like mild diarrhea or a cramp or flatulence, you know, or feeling, a little...
Which makes a lot of sense, right?
So, but what's really remarkable? We actually just published the results of this in the Journal for Immunotherapy Cancer, showing that the patients in that melanoma trial that did experience those side effects, the bloatings and the mild diarrhea and all that, they did better than the patients who didn't. So potentially that shows us that the microbiome is actually taking a foothold. The pharmacist is starting to crank up things.
Right.
And then the patient's experiencing it. It's not a serious side effect in terms of it's not grade 3 or grade 4 that would effectively hospitalize patients or require an intervention treatment. But it's enough that's telling us something's happening.
Right.
Okay. And I got to say, compared to what patients generally experience in oncology, with chemotherapy drugs or even side effects of immunotherapy, these are generally very mild side effects. But it's true, they do experience it. And mostly from what we have tracked so far, those who are doing really well actually experience it more than those who didn't.
That's very interesting.
Yeah.
It seems to me maybe like that week after taking the pills, there's a lot of information you're going to learn about what's actually like really happening in the body. Are you planning to look at that, or how much have you looked at that?
Yeah, so we have, and what we do normally for all the trials is that we take a sample from the patient right before FMT, and that is a stool sample, it's a blood, depending on trial, it could be urine. And then obviously we're also collecting clinical information from the patient. And then we, just after the week is done and before they start immunotherapy, it's very important that we are collecting that second sample before they start immunotherapy.
Right.
We're collecting samples from them, and then we're looking at all the changes, and we do see quite a bit of immune activation. For example, we do see changes in their metabolome. So we published in Nature Medicine before showing that histidine was a metabolite that's actually going up early on and stays up in all these patients. And we have done now another study that's under review following up on that, showing that it's actually doing maybe things that we think it's affecting the immune system in a positive way.
In a positive way, yeah, great.
So we are studying that, and that is very important to do because scientifically you want to establish mechanistically what's happening to the patient, because the reason this is important is that if we know what's happening in the patient, then we can potentially reproduce it in another patient.
Right.
If we don't know what's happening, then you don't know how you can actually reproduce it in another patient. And it's also very important to understand that, and that was something that was a learning experience for me as a PhD scientist. In science, we often have this reductionist approaches. So we take one factor out of the equation and test the hypothesis, and then we change another one in the lab because we're keeping all the conditions the same. But when you're translating that science or trying to translate that science into clinical medicine, into patients, into humans, now you're dealing with a complex system that's in front of you that's called the patient, okay? And you can't just control everything. So, and that's why the best science is the one that works across patients. That's why you have to understand the mechanism, try to establish that mechanism, and see if that is strong enough or important enough that can work in a patient who looks like you and a patient who looks like me, or a patient who looks like an 80-year-old female who is retired.
Yeah.
You know what I mean? So And that's why it's important for us to do the best science that we can to try to understand what's happening with these new treatments.
Amazing.
And that's part of the challenge with multidisciplinary trials as well.
Absolutely. I can see that. I think to kind of end our conversation, I know it's difficult for researchers in your field because you live in the moment. You know, you're one paper to the next and one experiment to the next. But are you able to take us 10 years into the future?
Mm-hmm.
Do you see a future? And what does that look like? And how is FMT and our gut going to play a role, 10 years down the line in potentially curing cancer or at least having effective treatments?
Yeah, so it's a C word in cancer. We don't often use it. We say for a patient who's stage 4 metastatic, they have cancer. So we try to help them to go into long-term remission and maybe make this a chronic condition. It's difficult to say
the C word is,
but having said that, there have been patients who've been treated with immunotherapy maybe 20 years ago now, and they're still in remission, and you can potentially say they are cured. What I got to say is that obviously as scientists, we are very focused on the science, and we go through this journey of trying to also publish our results for also the important purpose of informing the rest of the field. But what we get also out of this is that every time a patient responds, I get a text message from clinicians and they tell me that patient responded. And that actually makes a huge difference in terms of motivation for me, because I know I contributed to somebody's health. But what I see in 10 years, what I think we want to see the field goes, is that to try to understand the mechanism better, to understand when we are changing this microbiome in the patients with something like FMT. FMT, if you want to give it a weapon analogy, is like a nuclear bomb. Okay, right. So this is probably the strongest way you can change somebody's microbiome. You're taking their own biome, you're getting rid of their own biome and completely carpet bombing it with somebody else's biome. So that's a very strong approach, in fact. It's not as targeted, but it's very strong. So one of the things we are doing now is that we are trying to make that FMT treatment more tailored to the patient, making it more personalized. And I'm hoping that in the 10-year time, we have a scenario that we can effectively have a patient come to the clinic, do an initial microbiome test on that patient. It could be a blood test that just looks at metabolites, or it could be a microbiome test, look at the presence or absence of some key bacteria and say, okay, you're missing this bacteria or missing that key metabolite. We think based on this and the cancer you have and treatment you're going to get, this potential donor is the best donor for you.
Right.
And then match that donor to the patient. And we are also looking into maybe biological sex is a factor. Maybe if you're a female patient with breast cancer, you need a female donor. Or the opposite can be true as well. It could be that if you're a female, you may actually need a male donor because you want the microbiome to completely be different to upset the immune system in some ways and get it to go after the tumor.
Yeah.
So we are testing all those hypotheses.
Yeah.
So, and I think in 10-year time, we're hoping that FMT be more personalized, but I'm also sure that there's going to be more maybe tailored approaches that would come up, maybe more dietary support for patients who are getting FMT, or patients who may not need a microbiome intervention completely like FMT, but you can support what they have their own biome with a particular diet. Or even exercise. Actually, I'm collaborating recently with a group, Fred Hodge, in Seattle, and they're very interested in the role of exercise in seeing the change in the microbiome and preserving some of the functions of the microbiome. Maybe we can give you a particular set of exercises that helps support your healthy microbiome as you're going through the cancer journey. But what I'm sure is that this field will grow massively. And for the patients who are getting checkpoint inhibitors, those antibodies, or patients who are getting cellular therapy like CAR T therapy, or TCR therapy or TIL therapy, these are tumor infiltrating lymphocyte therapy, these are all parts of the cell therapy. You can maybe support their microbiome to get a better response, but also potentially reduce some of the side effects of their treatment. That's the other area that microbiome can be useful. So I'm hoping that we can have these microbiome-centric treatments as an adjunct to what we're giving the cancer patients to support their immune system and also potentially reduce some of the side effects they're experiencing, which is really problematic in cancer.
Yeah. This is so fascinating, and thank you so much for taking me on this journey. I'm so excited just, personally to see the next 10 years of what the gut's going to teach us about our bodies. On behalf of the team on the Catalysts, thank you for being here. Thank you for your persistence as a younger researcher, I guess in a sense, trusting your gut and not stopping and getting this treatment available for many, many people.
Thanks for having me. I appreciate it.
Yeah, great.
Thank you.
We hope you enjoyed this episode of The Catalysts, a medical science and discovery podcast by the Schulich School of Medicine and Dentistry at Western University, Canada, bringing you exclusive conversations with leading scientists and researchers.